Welcome to the Chemistry, Biology and Math Revision Hub. Today we're doing the Pearson Edexcel International A-Level Biology Unit 5 for October 2024. Let's begin with the first question. In question 1, neurons transmit impulses between receptors and effectors. The diagram shows a neuron. This is a sensory neuron. They say draw an arrow on the diagram to show the direction of nerve impulse. So it's going in this direction. This is the cell body. For a sensory neuron, the cell body is to the side, so it goes in this direction towards the central nervous system. Then they say nerve impulses travel along axons due to changes in membrane potentials. The graph shows an action potential in a neuron. We can see the different components of an action potential: the depolarization, repolarization, hyperpolarization, and then the restoration of the resting potential. They say which is the resting potential of this neuron. It's negative 70 millivolts. You can see at the beginning here it's minus 70 and then it's restored in the end. So minus 70 millivolts, which later on the graph shows where the neuron is hyperpolarized. Hyperpolarization is at W here. So the answer is C. And then which is the difference in membrane potential between points V and W in the graph. We have 150 and the other negative 90. The difference is going to be 140. They're talking about difference in magnitude, so 140 millivolts. Then give two differences between the functions of a motor neuron and a sensory neuron. This is for functions, not structure. The sensory neuron sends impulses from the receptor cells towards the central nervous system, while the motor neuron is going to come from the central nervous system towards the effector organs like muscles and glands. So I said motor neurons carry impulses from the CNS to effectors, muscles or glands, whereas sensory neurons carry impulses from sensory receptors to the CNS. Sensory neurons release neurotransmitters into the synapse, whereas motor neurons receive stimulation by neurotransmitters from relay neurons. In question 2, plants and animals use chemicals to coordinate metabolic activities and development, which is a function of gibberellins in plants. Gibberellins will stimulate the release or production of enzymes like amylase, so they do not hydrolyze, they do not inhibit elongation, they do not regulate translation. The answer is D. They regulate the transcription of genes like those leading to the formation of amylase. Then which is produced by an animal in a fight or flight response? This is adrenaline, and the answer should be B. Then in part B, tributyltin or TBT is a commonly used chemical in bodybuilding. The effect of TBT on the blood levels of two steroid hormones, testosterone and estradiol, was investigated. Groups of male mice were fed corn oil alone or corn oil containing different concentrations of TBT. The concentrations of testosterone and estradiol in the blood of the mice was measured. The results are shown in the graph. So here we see there is a decrease in the concentration of testosterone as the concentration of TBT increases. Error bars have been placed, but there is overlap. We can see this and this overlap. There is also overlap actually among these three. That overlaps with that and this overlaps with that, but there is no overlap at higher concentration. Then here we see the concentration of estradiol. This increases, peaks at 50, and then it decreases. Error bars have been placed, meaning repeat experiments were carried out, but still there is overlap until 50. So these results are not significantly different. So here they say describe 3 conclusions that can be drawn from this investigation. Like I've already described, increasing concentrations of TBT in corn oil leads to a decrease in testosterone levels in male mice. TBT exposure increased estradiol levels compared to the control until 50 milligram per kilogram TBT. However, at 100 milligrams per kilogram of TBT, estradiol levels decreased sharply. Testosterone levels were higher than estradiol. Higher concentrations of TBT, meaning 100 milligrams per kilogram, negatively affect both testosterone and estradiol levels. Then describe how steroid hormones regulate gene transcription. Steroid hormones diffuse through the cell membrane. They bind to specific receptors found inside the cells and they form a hormone receptor complex. This complex acts as a transcription factor binding to DNA and the promoter region, either to stimulate or inhibit gene transcription. In question 3, endurance athletes may suffer from muscle fatigue when contraction of the muscle fiber is disrupted. It is thought that the disruption of the contraction of muscles is caused by a lack of calcium ions within the muscle fiber. Explain why a decrease in calcium ions could disrupt muscle contraction. Calcium ions initiate the muscle contraction when they bind on troponin. So if there are no calcium ions, nothing is going to bind to troponin to cause the conformation change, which leads to the exposure of the myosin binding site. So I said fewer calcium ions bind to troponin. The shape of troponin is not changed, so tropomyosin doesn't move to expose the myosin binding site, and this prevents cross bridges from being formed and muscle contraction. Then they say the body controls the concentration of calcium ions circulating in the blood. This concentration is kept within the range 80 to 100 milligrams per decimeter cubed. Name the type of process involved in maintaining calcium ion concentration within this range. This process is homeostasis. Then the calcium ion concentration in the blood is kept within a range of 6% above and below the mean concentration. The mean calcium ion concentration in the blood is 91 milligrams per decimeter cubed. Calculate the maximum possible difference in the calcium ion concentration in the blood. Here they want us to find the maximum, the minimum, and then get the difference. So if 6% is the range within which we keep the concentration, so the deviation should be 6% of 91, which is 6 over 100 times 91, gives us 5.46. But this is a plus or minus, meaning to 91, when we add 5.46, we get the maximum value, and then when we subtract 5.46, we get the minimum value. So which are that, and then the maximum possible difference between the two values is going to be that minus that, giving us 10.92 milligrams per decimeter cubed as the answer. Then in part C, in an investigation, a group of cyclists completed a 30-kilometer time trial in a simulator. The blood lactate concentration was measured at rest, during exercise, and after exercise. The results are shown on the graph. Before exercise, the lactate concentration was lower since there is less anaerobic respiration. When exercise started, more anaerobic respiration took place, so the lactate concentration increased. However, they are trained and therefore their bodies have adapted to break down the lactate a little bit better. So as lactate is broken down into pyruvate, we see there is a decrease and then an increase like that. This fluctuation part is because the lactate is being broken down and then more lactate is produced. And then after exercise, we see the lactate concentration begins to go down because the person is not very active, and now the lactate is being broken down until it goes back to normal. Error bars have been included. There is overlap among here and this part and that part, so the results at this point and that are not significantly different, and the results at these levels are not that different. So they say explain the effect on the lactate concentration from 15 minutes to 60 minutes on the graph. This is when they are functioning at a higher peak. So I said from 15 to 60 minutes, lactate levels remain high and fluctuate slightly, meaning there is a little change due to anaerobic respiration during sustained exercise. Some lactate produced is converted to pyruvate. That's why we see the increase and decrease and increase and decrease like that. Then state one reason why the lactate concentration was measured 15 minutes before exercise. This is to get a baseline of the lactate concentration at rest so that we can compare that with the results when they are at peak activity or during exercise. Then state one variable that would need to be controlled in the investigation and how this would be achieved. So we could use people of the same age. The cycling has to be the same intensity. The area where they are should have the same oxygen availability or oxygen concentration. They could also have the same food intake. In question 4, Parkinson's disease is caused by a lack of specific neurotransmitters. The graph shows the effect of age on the number of dopamine producing cells in people who develop Parkinson's disease. So as age increases or as they get older, we see there is a decrease in the number of dopamine producing cells, meaning the number of patients with Parkinson's increases. In part A, they say in people without Parkinson's disease, the number of dopamine producing cells rarely fall below 350,000. Explain why the symptoms of Parkinson's disease increase with age. Use the graph to support your answer. As age increases, the dopamine producing cells are gonna die, and when they die, for example, at 44, this is where we have the 350,000, so this is gonna be the age limit with which we have that value, meaning people older than that, there is a high chance of them losing more dopamine producing cells and therefore getting Parkinson's. So I say the number of dopamine producing cells decrease with age. It falls below 350,000 after age 44. Fewer dopamine cells means less dopamine, fewer or no action potentials in motor neurons, and so there is less or no muscle contraction. In part B, the drug L-Dopa is used in tablet form to treat Parkinson's disease. This drug is converted to dopamine by an enzyme found in the digestive system and the brain. Carbidopa is an inhibitor of the enzyme found in the digestive system. Suggest why carbidopa is given to patients when they are treated using L-Dopa in tablet form. So it means this carbidopa is going to prevent the conversion of L-Dopa into dopamine. So it will ensure a higher concentration of L-Dopa crossing the blood-brain barrier. So I said carbidopa prevents L-Dopa from being converted into dopamine in the digestive system. Dopamine can cross the blood-brain barrier, but L-Dopa can't, so using carbidopa allows more L-Dopa to cross the blood-brain barrier. So less dosage of L-Dopa is given, and the effectiveness of the treatment increases. In part C, in an investigation, different concentrations of dopamine were given to Drosophila. The mean heart rates of groups of 10 Drosophila were recorded at different concentrations of dopamine, and the results are shown in the graph. Here they're using log to base 10 of the concentration of dopamine. We see as that increases, or the concentration or log to base 10 concentration of dopamine increases, the mean heart rate increases and then it decreases. Error bars are included, meaning repeat experiments were carried out. There is overlap here, within these two, so some of these results are not significantly different. So there's a comment on the results of this investigation. I said heart rate increases with dopamine concentration up to 4 mol per decimeter cubed. At the highest concentration, heart rate is going to drop like that, it drops. This suggests an optimal dopamine level for stimulation, because if the concentration is too much, the heart rate goes down, so there is an optimum. Error bars are included, but there is overlap, so the results are not significantly different for when there is overlap, for example here and there. Only 10 Drosophila were used. This is a small sample size, and there is no indication of their age as well as gender. In question 5, pronghorns are similar to antelopes and are native to North America. They live in open areas of grasslands. The photograph shows a pronghorn, which is that. They say these pronghorns can run long distances at speeds of 90 kilometers per hour. Which of the following features of pronghorns is an adaptation for running long distances at high speeds? They say decreased glomerular filter rate. No way. Increased number of slow-twitch muscle fibers. That's correct. It gives them endurance. Narrow airways. No. And small cardiac output. No. The answer should be B. This enables them to have endurance. Then explain how the heart rate and ventilation rate of a pronghorn are increased to enable it to run long distances at high speeds. This could be through the medulla involvement, meaning the nervous system is going to stimulate the heart, more so the sinoatrial node, to cause more depolarization of the heart muscle, as well as more depolarization of the lung muscles like the diaphragm, as well as the intercostal muscles. So I say during exercise, the blood pH decreases due to carbon dioxide increase as well as lactic acid. This decrease in pH is detected by chemoreceptors in the aorta and carotid arteries. Impulses are sent through the sensory neurons to the medulla oblongata. A response to the sinoatrial node, intercostal muscles, and the diaphragm cause more depolarization. The sinoatrial node sends more excitation to the heart muscle to increase contraction and get rid of carbon dioxide, while the intercostal muscles and diaphragm cause more frequent contractions for inhalation and exhalation to remove the carbon dioxide. In part B, scientists measured the mass, resting metabolic rate, and pulse rate of 5 mammals. The table shows the results. Here we see the size of the mammal increasing, or we can say the mass. As the mass increases, the resting metabolic rate also increases, but the pulse rate decreases, as we can see there. So they say describe the relationship between mass, metabolic rate, and pulse rate shown in the table. Like I've said, as mass increases, the resting metabolic rate also increases. However, pulse rate decreases with increasing mass. Also, as resting metabolic rate increases, pulse rate decreases. When a human exercises, the metabolic rate is increased and heat energy is released. Describe how the body temperature of a human is maintained at 37 degrees Celsius during exercise. This is thermoregulation involving thermoreceptors, the hypothalamus, and organs like the skin. So I said thermoreceptors in the skin and hypothalamus detect a rise in body temperature, meaning above the optimum during exercise. They send impulses to the thermoregulatory center in the hypothalamus. The center then sends signals to effectors like the skin, where we see the sweat glands, blood vessels, causing sweating and vasodilation to increase heat loss and bring the temperature back to normal. Impulses could also be sent to organs like the liver to decrease the metabolic rate so that less heat is generated in the body in order for this homeostatic mechanism to return the body temperature to normal. In question 6, the kidney has an important role in the regulation of the plasma concentration and blood volume. The photograph shows the Bowman's capsule, which is the renal capsule. The line from X to Y shows the diameter of the Bowman's capsule. So they say the diameter from X to Y is 70 nanometers. Calculate the magnification of this photograph. So you can use a ruler and measure that line as well, or the length. XY is about 52 millimeters, which is 52 times 10 power 6 nanometers. So the magnification is going to be the image divided by the actual. So image is this divided by the actual, which is 70. And so I got 742,857, which is roughly 7.43 times 10 power 5 as the answer. Then next, calculate the volume of this Bowman's capsule. Assume that this Bowman's capsule is spherical. They want us to use this formula. Since we know the actual length is 70, that is going to be the diameter, so divide that by 2 to get the radius. So the volume will be 4/3 times pi times the radius, which is 35 power 3, giving us that. But the answer should be to two significant figures, so we round off to the closest, giving us 180,000 nanometer cubed. Then part B, which is the specialized receptor that detects changes in the plasma solid concentration. This is going to be osmoreceptors. We're talking about concentration. These are for pressure. These are for, of course, pH and chemicals. This is for temperature changes. The answer is a C. Then suggest one reason why the plasma solid concentration of the blood needs to be kept within a narrow range. If it's not kept within a narrow range, then the cells are going to be affected. For example, if the concentration in the plasma is higher, cells are going to lose water by osmosis. And if the concentration is lower, the cells are going to gain water by osmosis, and the cytoplasm is going to be changed, meaning reactions taking place inside cells are going to be affected. So I say to maintain water potential for proper cell function and prevent cells from bursting or shrinking due to osmosis. In part C, drinking too much water causes the plasma solid concentration to decrease and the blood volume to increase. Describe how the body responds to this change. When the blood volume increases, meaning there is much water, ADH will not be released, and so more urine is going to be produced. But when the blood volume decreases, the pituitary gland is going to release ADH, which is going to cause the kidney tubules, like the distal convoluted tubules, as well as the collecting ducts, to reabsorb more water in order to return the blood concentration to normal. So I say the change in plasma concentration is detected by osmoreceptors in the hypothalamus. Blood volume change is detected by baroreceptors in the aorta and carotid artery. Impulses are sent to the pituitary gland and less ADH is released. Less ADH will be released if there is increase in blood volume. So less water is reabsorbed in the distal convoluted tubules and collecting ducts of the kidneys. More dilute urine is produced, the blood volume and solid concentration returns to normal. In part D, which method reabsorbs glucose from the proximal convoluted tubule into the blood? This is going to be co-transport with sodium. The answer is A. These membrane proteins will facilitate the movement of sodium as well as glucose together. The Bowman's capsule of a healthy kidney filters up to 7.5 grams of glucose per hour. Calculate the rate of glucose filtration in milligrams per second. So in 1 hour, we have 7.5 grams. I'm converting this to milligrams is 7.5 times 10 power 3. In 1 hour is 3600 seconds. So in 3600 seconds, this is the amount. And in 1 second, it's going to be that divided by that, giving us 2.083. And that's the answer. In question 7, some diseases and certain cancers are thought to be caused by a lack of specific neurotransmitters. This means they can be treated using drugs which increase the concentration of these neurotransmitters. Some of these drugs are manufactured using genetically modified organisms, which is a description of a GM organism. A genetically modified organism is one containing genetic material from another species. So the answer is B. Then they say BVZ is an antibody used to treat brain cancer patients. This antibody is produced using animal cell cultures. A new method of producing BVZ has been developed using genetically modified rice. The graph compares the antigen binding activity of a BVZ antibody produced using a control using non-transformed rice, animal cell culture, 4 strains of GM rice A, B, C, and D. So we see the results. In this non-transformed, we can see it has the lowest. Then the highest is in the animal culture. Then among the strains of rice A, B, C, D, we have the highest in B and the lowest is going to be in D, although it's not that different from this one here. Error bars have been included and there is no overlap, so the results are significantly different. They say describe 3 conclusions that can be made from these results. So all samples produced BVZ, however, animal cultures produced BVZ with the highest antigen binding activity compared to GM rice in non-transformed rice. Strain B of GM rice produced BVZ antibody with the highest binding activity close to that of animal cultures. Non-transformed rice produces very low antigen binding activity, indicating it is not effective at producing functional BVZ. Then explain how BVZ antibodies can cause the destruction of cancer cells. These are antibodies, so they're going to be specific and they will bind to specific receptors on cancer cells. So I said BVZ antibodies are complementary to receptors on the surface membrane of cancer cells. They bind specifically to antigens on the surface of cancer cells. This marks the cell for destruction by phagocytosis or by T killer cells. In part C, maize is a major food crop. The photograph shows a maize cob with rows of grains. We can see the grains. Maize can be infected by a fungus. This fungus produces a toxin that affects plant growth and is poisonous to animals. The fungus can be genetically modified. In an investigation, maize plants were infected with normal fungus or infected with GM fungus. The table shows the mean and standard deviation for growth measurements of maize and the concentration of toxin. So they've given us for normal fungus and GM fungus, the plant height, the number of grains per cob, and the concentration. Infected with GM fungus, these are going to be taller in comparison to those. And these ones here, the GM ones, will have more number of grains per cob. And here we see that concentration of the toxin is going to be more in the normal. The deviations have been included and there is overlap, as you can see in some of them. So they say a computer model was used to predict the effect of global warming on the percentage of maize crop infected with this fungus. The results are shown in the table. So the current temperature, we see the percentage of maize corn predicted to be infected is 38. When the temperature increases by 2 degrees, we see this increases to almost doubling. And if it's increased by 5 degrees, its increases were higher. So as temperature increases, the percentage of maize crop predicted to be infected by the fungus is increasing. So as global warming occurs, it means more grains are going to be affected by the fungus and maybe there will be less food that is available. They say assess the risk and benefits of genetically modifying the fungus for future food production in a globally warming world. Use the information given in the table together with your own knowledge to support your answer. So since I've already gone through the tables, I'll just read what I wrote. I say genetically modifying the fungus that infects maize could offer major benefits for food production, particularly as global temperatures continue to rise. The data shows that maize infected with GM fungus grow taller and produce slightly more grains per cob compared to maize infected with a normal fungus. The GM fungus leads to a significantly lower concentration of toxin in the crop, making the food safer for both humans and animals. As global warming intensifies, fungal infections are expected to become more widespread, like we see from this table here, raising from 38% of the crop currently to 95% with 5-degree increase in temperature. This makes reducing the harmful effects of infection even more crucial. Since the GM fungus produces less toxin, it could help ensure that even if the infection rates increase, the impact on crop safety and yield is reduced. Healthier, more productive crop means farmers can achieve higher yield without needing to expand farmland, and this is going to reduce pressure to clear forests and other natural habitats for agriculture, helping to preserve biodiversity and protect ecosystems. Efficient land use also lowers the carbon footprint of food production, especially if more food can be grown locally and there is less reliance on transport or imports. These factors contribute to better food security and could help reduce malnutrition, particularly in vulnerable regions. However, there is a potential risk. Using GM fungus on a wide scale may lead to reduced biodiversity through monoculture farming. There is also a risk that the modified genes could spread into other organisms, possibly creating new problems like superfungi. Some people view genetic modification as unnatural, and there are ethical concerns around companies owning the technology and potentially limiting access to poor farmers. Additionally, the long-term health effects of consuming GM crops are still not fully understood. It's also important to note that while the toxin levels are lower with GM fungus, there is still some overlap in the data, meaning the difference may not be statistically significant. To conclude, GM fungus offers promising benefits in reducing toxins and maintaining crop yield in a warming world. However, careful monitoring, transparent regulation, and long-term studies are essential to minimize risks and ensure the technology is used responsibly. So now we can begin question 8. This is a scientific article you have studied is adapted from articles about Alzheimer's disease. Use the information from the scientific article and your own knowledge to answer the following questions. The first says, describe the possible role of hypertension in causing dementia. Because hypertension causes damage to blood vessels, this could occur in blood vessels leading to the brain. And so if there is damage, there is less oxygen taken to the brain, and so the brain cells could die, leading to dementia. So I said hypertension can damage blood vessels in the brain, reducing blood flow. This leads to less oxygen and nutrients reaching brain cells, causing cell death, contributing to dementia. Then in part B suggests why the protein beta amyloid from the fatty membrane surrounding nerve cells is chemically sticky, gradually clumping together. Use your knowledge of protein structure to support your answer. Proteins have hydrophilic and hydrophobic R groups, but in this case, due to problems in folding of the proteins, the hydrophobic R groups are folded on the outside, creating clumps that form plaque. So I said beta amyloid proteins are fat soluble, meaning they have hydrophobic R groups. Misfolding of proteins can expose the hydrophobic R regions, making them attract each other, being sticky, and these sticky regions cause proteins to clump together, forming plaques. Describe the information that will be obtained from using a combined MRI and PET scan in the brain. MRI is going to show detailed structure of the brain, including shrinkage or abnormalities. And the PET scan shows brain activity by detecting areas of high or low glucose metabolism. Then state one reason why it might be considered unethical to tell people they are at risk of developing cognitive impairment. It may cause emotional distress or anxiety, especially if no cure or effective treatment is available. Explain why a falling blood pressure may result in poor brain health. If there is low pressure, then less glucose and oxygen is going to be delivered to the neurons. And so the neurons are going to be affected, they'll be damaged. So I said lower blood pressure can reduce blood flow to the brain. This results in less oxygen and glucose delivery to neurons, so less aerobic respiration. Neurons may become damaged or die, impairing brain function. Then some genes may be linked to an increase in the risk of dementia. Describe how scientists could identify the link between these genes and the risk of dementia. You could use microarray to compare the DNA of people with and without dementia. Then identify genes or alleles that are more common in people with dementia. They should be together. This helps to identify people with dementia in their family history. Then perform statistical analysis to see if these genes significantly increase risk. Then describe what is meant by the term monoclonal antibody in paragraph 13. These are antibodies that are produced from a single plasma cell, or you say the same cell line. They bind to one specific antigen, causing agglutination. Give one reason why a placebo was used in the donanemab trial, paragraph 33. A placebo acts as a control to ensure that the observed effects are due to the drug and not due to the people's psychological effect because they took the drug. Then swelling is a symptom of inflammation. Explain why lecanemab may result in brain swelling. It triggers an immune response as it clears amyloid due to release of histamine. This inflammatory response can increase fluid in the brain tissues, causing damage to blood vessels. Then successfully clearing amyloid from the brain had only a small effect. So just one reason why researchers are suggesting the need to start using the drugs even earlier in the disease. This is because the symptoms take longer to appear, and if we use the treatment earlier, then it's going to slow down the progression of the disease. So I say the brain damage from dementia occurs before symptoms appear. Earlier treatments may prevent or slow disease progression more effectively. So this brings us to the end of question 8 as well as to the end of the whole paper. Thank you for being with us. Do not forget to subscribe. See you in the next video. Bye-bye.